review of DNA protein synthesis with more in-depth details on transcription. First, the unzipped DNA serves as a template for the messenger RNA to be created by RNA polymerase, bonding A's to T's, C's to G's, but U's to A's.
Jeez.
So RNA only has uracil versus thymine. Okay. then the messenger RNA is sent to the ribosome, and from there, the specific sequence of amino acids creates the polypeptide with the help of tRNA and rRNA, and that determines the role of the cell in DNA synthesis of proteins.
And go.
First of all, DNA is the
I'm leaving that in there.
Nucleotides are the building blocks of nucleic acid polymers. The basis of DNA are thymine and cytosine, also known as pyrimides. Purines are adenine and guanine. DNA strands are bound by covalent bonds. There's a repeating pattern. Yeah. Double helix structure, found out by Watson and Crick. Sugar and phosphate backbone. Adenine marries thymine.
Guanine elopes with cytosine. They're both complementary.
Hershey and Chase devised an experiment on whether DNA or protein directs DNA to replicate. What? One batch of how many batches? One batch! One batch! One batch! Infected sulfur to label on the Let's go! Okay. Used radioactive phosphorus to label the DNA. Allowed each to infect separate cultures of bacterial cells. Concluded DNA was a hereditary material.
Yeah.
Doorknob.
Avery, McLeod, and McCarthy tested whether protein was transforming factor.
Man, they smart.
Treated Griffith's heat-treated deadly strain and live harmless strain with protein-destroying enzymes. Bacterial colonies were transformed still. Protein was not transforming factor, then treated with DNA-destroying enzymes and
What are you saying? I wrote shorthand, you're supposed to...
and colonies failed to transform. Conclusion again, DNA is genetic material of cell. There you go.
Traces of information flow from DNA to RNA to proteins. RNA contains uracil instead of thymine. It pairs with adenine instead. DNA sequence is converted to a single strand of RNA. That's pretty much what transcription is. Translation is nucleic acid language being converted to the amino acid language. And then a codon is a three-base word that codes for one amino acid. Several codons form a sentence.
Here it goes.
Yeah.
Mutation is any change in nucleotide sequence of DNA. Insertion or deletion of more than one nucleotide of a gene is more disastrous than a base substitution. Mutagens are physical or chemical agents that cause mutations. One type of physical mutagen is high-energy radiation. Get up! X-rays and ultraviolet light are high-energy radiation.
Oh yeah?
Yeah. Look at these butterflies. Hold on.
That's not a...
Look at these butterflies. That is genetic mutation. They're the same species, but they look completely different.
DNA replication. Let's say this is a strand of DNA. Helicase will come and break the hydrogen bonds between the nucleotides to split the DNA strand. Then, DNA polymerase will bind to specific parts of the DNA and bind nucleotides to the pre-existing nitrogenous bases, binding A's to T's and C's to G's in a 5' to 3' direction. Then, after the strands are finished being copied, you end up with 2 strands of DNA, each with 1 parent strand, and a completely new strand of DNA. Now what does all this mean, you might ask? I'll tell you. America.
By the din of fairyland, his day is marching